Hey everybody, welcome back to my podcast, Anatomy and Physiology Bit by Bit. This is your host, Dr. Steve Sullivan from Bucks County Community College, outside of Philadelphia, Pennsylvania. This episode, we're going to finish up the cell, and we're going to do that by talking about the way a cell will make a copy of itself, or duplicate, by division. That means the cell will divide into two new cells, so that we can either grow or maintain our tissues. This is an important concept because we utilize this particular process for a lot of different things. Again, I mentioned to grow, to get bigger, or to develop from the embryonic stem cells that start our life into the trillions and trillions and trillions of cells that we have now as a fully formed human. So there's a lot of things in addition to that maintaining our cells. Cells don't live forever, so when cells die or they outlive their usefulness, they get replaced. Also healing from an injury or recovering from an illness. All of that is utilizing our cell's life cycle, specifically division, which would be division of the nucleus or mitosis, and division of the cytoplasm, or cytokinesis. The interesting thing about the way we do things in our bodies is that we multiply by division. I know it sounds kind of weird, right, to say it that way, but in order for us to multiply the number of cells in our body, we have to divide those cells. So one cell divides into two cells divide into four cells divide into 8, etc. So that's kind of how we do it, and we're going to use this episode to get into exactly how that happens, because in order for us to do that, we need to copy our genetic material so that each cell is identical. And I'm going to discuss that. We call that DNA replication. So that's what we're going to start off with. We're going to start off with DNA replication. Before I get into that, I just want to mention a couple of quick things. Some of you may have been curious about my cat. I mentioned my cat a few episodes ago. He had hyperthyroidism. He was treated with a radioactive isotope of iodine, and he was quarantined for 2 weeks because of that radioactivity. And I've gotten a couple of emails from people sending well wishes, which is really nice. I really appreciate the support and the feedback and the well wishes for my 15-and-a-half-year-old cat, Getty. Yes, he is named after Geddy Lee, the bass player and lead singer of the band Rush, who lost their drummer in January of 2020. Well, he's doing great, by the way. My cat is doing really well. He's out of quarantine. He's allowed to roam around the house and sleep in our room and play with the other cats and the kids and all that stuff. So for those of you who were concerned and asking and sending well wishes, Getty the cat is doing fantastic, so I really appreciate it. I think he's got a few more really good years in him. He's a good, healthy cat. So thanks again for all those well wishes. All right, so like I said, we're going to tackle the remaining piece of cell bio that's important for anatomy and physiology. So if you're taking an anatomy and physiology 2-semester course in undergrad, this is about the level you're going to need for this particular course. If you're just listening because you're interested, fantastic. I think this stuff is interesting too. Okay, so why don't we get going with DNA replication? I hope you enjoy it. Let's move. The life cycle of the somatic cell. It consists of interphase and mitosis. Interphase is when a cell is not dividing, and cell division includes two different phenomena: cytokinesis, which is the division of the cytoplasm, and mitosis, which is the division of one nucleus into two identical nuclei. In order for a nucleus to do that, the DNA must be copied so each nucleus gets a full version of the original. So in this video, let's take a look at interphase, specifically DNA replication. Interphase has 3 phases of its own, 2 gap phases and a synthesis phase. The first gap phase, which is called G1, is when the cell grows and goes about its normal metabolic activity, like protein synthesis. The synthesis phase, which is also known as the S phase, is when DNA replication takes place. And the second gap phase, G2, is when the cell continues to grow and prepares itself for mitosis. Let's focus on the DNA replication part of the S phase. We'll call the original DNA the parent DNA, and the two new ones the daughter DNA. The first thing that happens is the core particle of DNA disassembles, freeing up the double helix from the histones. Then an enzyme called DNA helicase breaks the hydrogen bonds between base pairs, opening up a segment of the double helix like a zipper. The point at which the double helix opens is called the replication fork. Next, multiple enzymes called DNA polymerases on each strand of the original DNA molecule bring free DNA nucleotides to bind with the newly opened nitrogenous bases. Because of complementary base pairing, we know that each new bond will be specific, and it will match the bond that was there in the parent DNA. This will form two identical copies of the original double helix. Between the DNA polymerases is an enzyme called DNA ligase. This closes the gaps between polymerases. Each daughter DNA has one newly formed strand and one strand conserved from the parent DNA. For that reason, this is called semi-conservative replication. Once this is complete, new histones formed in the cytoplasm will be available to reassemble the new DNA into its original configuration. At this point, the nucleus is prepared to divide into two genetically identical nuclei, each with a full copy of the cell's original DNA. It's time to talk about mitosis. Mitosis is the division of the somatic cell's nucleus, creating two identical nuclei in preparation for cell division. The dividing cell is called a parent cell, and this process is necessary because each new cell, called a daughter cell, will need a complete nucleus to function properly. Mitosis is necessary for multicellular organisms like humans to grow and repair our tissues. So let's take a look at how mitosis works. The word soma is Greek for body. Therefore, somatic cells are the cells of our body. They include everything except the reproductive cells that give rise to sperm cells and egg cells. Except for neurons and cells with no nucleus, most somatic cells undergo mitosis. Let's start with a non-dividing nucleus. The genetic material in a non-dividing nucleus consists of DNA and proteins, and it exists in a thinly dispersed orientation called chromatin. A human somatic cell has 22 different chromosomes plus the X and Y chromosomes that determine biological sex. The number of different chromosomes in a cell is called its ploidy. So human ploidy is 23. Since each of our parents donate a set of chromosomes to us, each human somatic cell has a total of 46 in its nucleus. A cell with two sets of chromosomes is called diploid, for 2 times the ploidy. A cell that has only one set, like an egg cell or a sperm cell, is called haploid. Since the DNA replicates during the S phase of interphase, by the time we get to mitosis, each of our 46 chromosomes is comprised of 2 identical sister chromatids. Those chromatids are bound to each other by a structure called the centromere, and each centromere is wrapped by a protein called the kinetochore.
You buy a pair of socks, that's 2 socks. You buy a pair of Bombas socks, that's 4 socks. Because one purchased is one donated. Socks are the #1 most requested clothing item in homeless shelters. So when you buy a pair of super comfortable Bombas socks, you're also donating a pair. Bombas customers have powered over 150 million donations. So Bombas would like to thank you 150 million times, but we only have like 30 seconds. Go to bombas.com and use code AUDIO for 20% off your first purchase. That's BOMBAS.com and use code AUDIO at checkout.
Division of the nucleus, called mitosis, takes place in 4 phases: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes of chromatin condense and shorten. The nuclear membrane disintegrates and disappears, and the centrioles begin to sprout microtubules called spindle fibers. As these spindle fibers get longer, they push the centrioles to opposite poles of the cell. Some of these spindle fibers end up attaching to the kinetochores of the chromosomes. In the next phase, called metaphase, the spindle fibers tug on the kinetochores, pulling them to the midline of the cell, called the equatorial plane. The spindle fibers collectively form an oval shape called the mitotic spindle, and short microtubules from the centrosomes form the asters, which anchor the mitotic spindle to the internal surface of the plasma membrane at the poles of the cell. During the next phase, which is called anaphase, an enzyme splits the sister chromatids from one another at the centromere. Each chromatid is now considered a separate, individual chromosome. Each of these will migrate to opposite poles of the cell as motor proteins in the kinetochore crawl along the spindle fiber. The last phase of mitosis is called telophase. During telophase, the chromatids cluster on each side of the cell as the rough endoplasmic reticulum produces a new nuclear envelope around each cluster. The chromatids begin to uncoil and return to the thinly dispersed chromatin form. Now, while anaphase and telophase are happening, what's also going on is cytokinesis. This is division of the cytoplasm. So not only do the nuclei need to divide into 2 identical nuclei, but we also need to replicate the organelles of the cytoplasm and split the plasma membrane into 2. That creates the cleavage furrow. This is achieved by a protein pulling on microfilaments in the terminal web of the cytoskeleton. And the cell pinches in 2, creating 2 identical diploid daughter cells, each identical to the parent cell. Okay, that's interphase, DNA replication, mitosis, cytokinesis, all right there, the end of our series on the cell. The next episode, we're going to move into tissues, or histology, and then we're going to keep moving on through there. I hope that you enjoyed this. I hope you learned something from it, and I really hope it's helped you get a B or better on your next A&P exam. Good luck. Talk to you next time. Hey, don't forget to check out my YouTube channel, Student Help for AP. That's the word Student Help, the number 4, AP. And I also have an Instagram account and a Twitter feed with the same name. So check those out too. Anatomy and Physiology Bit by Bit is a production of Minus 55 Media, with a special thanks to Bucks County Community College for giving me a job doing what I love.